Okay, so welcome back to this next video in which we are discussing asthma and anti-asthmatic drugs. Okay, so we've now discussed the core things that underlie the airway hyperresponsiveness in both atopic asthma and non-atopic asthma. Okay, so we've discussed that what occurs is this chronic allergic response involving T helper 2 cells, okay, which invade into the lamina propria of the airways, okay, and release cytokines, principally interleukin 4, interleukin 5, and interleukin 13. The ones that we've seen that are important so far are interleukin 5 and interleukin 13. Okay, these cause this airway eosinophilia, this recruitment of a huge number of eosinophils into the lamina propria, and then interleukin 5 activates the eosinophils. The eosinophils then start releasing a huge number of cytotoxic proteins and also the growth factor, transforming growth factor beta. The cytotoxic proteins kill columnar epithelial cells lining the airways and therefore result in the production of holes in the airway epithelium. The dying columnar epithelial cells start releasing a lot of transforming growth factor beta, so we get even more TGF-beta being produced. Okay, the transforming growth factor beta then acts on the airway smooth muscle cells and causes hypertrophy and hyperplasia, which results in the mass of the bronchial smooth muscle cell layer around the bronchi increasing. Okay, and therefore its ability to constrict the bronchi goes up. Okay, right. So how does this then lead to airway hyperresponsiveness? Well, at the moment we're just talking about the non-allergic asthmatic attacks. So asthmatic attacks induced by cold air or noxious gases, okay? And those now can activate mast cells because of the holes in the columnar epithelium. So they have a better ability to activate mast cells in the lamina propria. And we will be discussing mast cells in a huge amount more detail in a moment. Okay, the mast cells are then the cells which, once activated, send the instruction to the bronchial smooth muscle cells to contract and therefore constrict the bronchus. Okay, so that's how these stimuli can now cause bronchoconstriction in asthmatics, whereas they couldn't cause them in non-asthmatic people. But in addition to that, you've also got to take into account that now the bronchial smooth muscle cell layer is much thicker, and therefore the actual amount of bronchoconstriction that you're going to be able to produce is going to be increased as well. Okay, so that's then the two things that underlie the airway hyperresponsiveness: the holes in the columnar epithelium and the increased bronchial smooth muscle cell mass. Okay, so what we now want to look at then is what in addition occurs in atopic asthma, okay, which allows you to have allergic asthmatic attacks, allows these mast cells to be activated by the allergen itself. Okay, and then what we want to do is actually study the mast cells in much more detail and how they're activated, and when they're activated, what they then produce, which is the stuff that sends the instruction to the bronchial smooth muscle cells to constrict. Okay, so firstly, let's start off then with how you can get allergic asthmatic attacks in atopic asthma. Okay, so what in addition happens in atopic asthma? Well, basically, in atopic asthma, we know that what happens is you get B cells activated against the allergen, okay, not just T cells. Okay, so we've seen how CD4 positive T helper 2 cells are activated in both atopic and non-atopic asthma against this allergen that you're being chronically exposed to. In atopic asthma, but not in non-atopic asthma, you also get B cells being activated against your allergen, and we will discuss this in a lot more detail later on. Okay, these B cells are then going to produce you plasma cells, which can produce antibodies against your allergen, basically. Okay, now that other cytokine released by T helper 2 cells, we didn't explain what it did previously, which is interleukin 4. Okay, this drives B cells to produce plasma cells, which produce a specific type of antibody molecule known as IgE. Okay, so IgE stands for immunoglobulin E. So an immunoglobulin is another name for an antibody molecule. So antibody and immunoglobulin, they're exactly the same thing. Okay, let me just explain what the E actually means. What is the difference between IgE and other antibody molecules? Okay, right, so for this I need to draw the structure of an antibody molecule out. Okay, so antibody molecules are made out of 4 proteins stuck together, basically. Okay, 2 what are known as heavy chains, okay, which are these long lines that I have here. So this line here and this line here, those are representing the heavy chains of the antibody molecule, or the immunoglobulin heavy chains as they're called. Okay, and I'll just abbreviate immunoglobulin down to Ig, like so, which is conventional. Okay, then you also have 2 immunoglobulin light chains here, which are these smaller ones. Okay, now basically, we are going to be producing antibody molecules which are targeted against an epitope of our allergen. So if I bring up that picture of our allergen again, okay, when we initiate a B-cell response, we're going to be selecting B cells that produce antibody molecules that can bind to a certain epitope of this allergen. Okay, and we might be activating multiple B cell responses. Okay, we might be activating different B cells that have antibodies capable of binding to different epitopes of the allergen. But we are at least activating one type of B cell that has an antibody which can bind to one epitope of this allergen. Okay, now which portion of the antibody molecule is it then that is specific for binding to a certain epitope of the allergen? Okay, well that's the variable region here. So the portion that I'm now colouring in red here, this is the portion that is specifically targeted against that epitope of that allergen. So this is the portion which is capable of binding to another protein, okay, and it will be specific for our allergen, so it will be directed against our allergen. Okay, so this is what's known as the variable region, okay, because obviously the adaptive immune system is capable of producing B cells that make antibodies against a huge number of different proteins, and all of their different antibody molecules will have different variable regions, and that's how they can bind to so many different proteins. Okay, so the antibodies that we are specifically interested in here then have variable regions which are capable of binding to an epitope of our allergen. Okay, now the variable region of an antibody molecule has another name. It's also known as the FAB. Okay, and what this stands for is the fragment, that's the F, and then the AB is for antigen binding. Okay, so normally when we're talking about immunology, we're talking about antibodies that are actually directed against a protein that should have an antibody directed against it. Okay, and those sort of proteins are called antigens. Okay, we're obviously discussing an allergic adaptive immune response, so rather than calling our protein an antigen, we're calling it an allergen. But effectively, antigen and allergen, as far as antibody binding to them is concerned, they're the same thing. An antigen is just any protein which can have an antibody produced against it. Okay, so obviously allergens are antigens, but not all antigens are allergens. Some antigens do deserve an immune response being initiated against them. Okay, right. Now, that's the portion then that varies between different B cells. So different B cells will produce antibodies with different variable regions. Okay, but this other portion down here is the portion that we're now interested in explaining what is meant by IgE that is directed against our allergen. Okay, this is what's known as the constant region. Okay, so this is called the constant region, or the fragment crystallizable is the other name for it. So it's also called FC for fragment, and then crystallizable because it will crystallize. Okay, right. Now, although this portion is called the constant region, there are actually different constant regions that you can use, basically, when you produce an antibody molecule. So if a B cell wants to produce an antibody molecule, it has a special variable region that it is fixed in making. Okay, and that's the portion that is directed against a certain epitope of an antigen. Okay, the constant region then, it actually has a choice which one it wants to use. Okay, there are many different constant regions it can use. Okay, and these different constant regions will have different structures and they can interact with other things. So once the variable region is actually bound to your antigen, okay, what does the immunoglobulin molecule actually do? Well, that will depend on what this constant region here is. Okay, so this is what the difference between the different types of immunoglobulin are. IgG, IgA, IgM, IgD, and IgE. This is what this is referring to. All of these different types of immunoglobulin molecules, it's referring to a different type of constant region of the immunoglobulin molecule. Okay, so in IgE molecules against a certain epitope of our allergen, it means that the variable region is, you know, just that portion that binds to the epitope of the allergen, whatever that may be. But then this constant region, we have selected IgE constant region basically, and that then produces us an antibody molecule which is called an IgE molecule. And these IgE molecules against that certain epitope of the allergen are capable of causing certain things. Okay, so that's what this is all about. Basically, if you have B cells maturing into plasma cells in the presence of interleukin-4, it tends to cause those plasma cells to, in the end, produce antibody molecules that are of the IgE type. Okay, now IgE is not the main type of antibody molecule to produce. IgG is the usual one that plasma cells end up producing. Okay, but if your B cells are maturing in the presence of interleukin-4, it can drive the production of plasma cells, which then produce antibody molecules of the IgE type, and plasma cells are effectively factories for antibody molecules. They'll chuck out a huge number of antibody molecules. Okay, so they'll chuck out loads and loads of IgE molecules that are directed against a certain epitope of the allergen basically. Okay, right, so in atopic asthma but not in non-atopic asthma, you get this B cell response which produces you plasma cells which will produce IgE molecules where the variable region is targeted against some epitope of your allergen. Okay, now what's the significance of this? Why does this then lead to these people with atopic asthma being able to have allergic asthmatic attacks? Well, basically, on the surface of the mast cells, you have a receptor that can bind IgE molecules. Okay, so if I draw a little picture, okay, now for the first time I'll actually show you a picture of what a mast cell looks like. Okay, so a mast cell is a typical circular cell, like so. Okay, here's its nucleus, and its cytoplasm is full of granules. Okay, and these granules contain a really important molecule. These granules contain one of the molecules which gives the order to smooth muscle cells to contract, basically. Okay, so remember, once the mast cells are activated, they send the order to the smooth muscle cells surrounding the bronchi that they need to contract. Okay, what is the actual molecular form of that order? Well, one of the key molecules that is that order is histamine. Okay, and the histamine is already synthesized and stored in these granules within the mast cell, basically. So here in green, these are the histamine granules. And basically, once the mast cells are activated, they'll degranulate and release the histamine into the extracellular fluid. It can then go and act on these smooth muscle cells and cause them to contract. Okay, so here is a picture of a mast cell then. Right, so these are the cells which can be activated, it's believed, by cold air and by noxious gases to cause non-allergic asthmatic attacks, okay? But to cause allergic asthmatic attacks in atopic asthma, they have a receptor on their surface called the Fc epsilon R1 receptor. Okay, and we'll be discussing this receptor in a lot more detail in a moment. Okay, this stands for the fragment crystallizable. Okay, so the constant region, epsilon is the Greek equivalent of E, so this means the Fc region of an IgE molecule, receptor 1, and basically that is just a receptor which can bind to the constant region of IgE molecules. Okay, so here now is our IgE molecule, but it can't bind to the constant region of different types of immunoglobulin molecules. Okay, so when you produce the IgE against your allergen, what happens is the IgE ends up in the lamina propria, attached to these mast cells in the lamina propria. And I don't know if I stressed that enough. The mast cells are in the lamina propria. Okay, so I'll underline them in blue since I coloured in the lamina propria earlier in blue. Okay, right. So in atopic asthma then, you produce IgE against an epitope of your allergen. This IgE ends up mounted on top of these special receptors on the surface of the mast cell here, okay, which are Fc epsilon R1 receptors, okay, and this now makes the mast cell armed against the allergen, because now if you inhale the allergen, if you inhale a very large dose of the allergen then comes into your airways, it crosses the airway epithelium, potentially through the holes that you've now got in the airway epithelium, gets to the mast cells, binds to the IgE that's specialized against a certain epitope of the allergen, and that then activates the mast cell, just like cold air and noxious gases could activate the mast cell. The mast cell then releases histamine and other molecules, which are these orders going to the bronchial smooth muscle cells to contract, and therefore you get bronchoconstriction, which is the main part of the asthmatic attack. Okay, right. So now what we want to discuss is asthmatic attacks in more detail. Okay, so firstly, what I want to discuss is that there are two phases of asthmatic attacks. Okay, so I'll just write this down here. So asthmatic attacks, they can be divided up into two phases. Okay, there is the immediate phase, which occurs very quickly after the mast cells have been activated. Okay, and the immediate phase involves the actual bronchoconstriction. Okay, so the immediate phase is the scary phase, where the bronchi and bronchioles are constricting because the bronchial smooth muscle is contracting under the orders of these mast cells. Okay, then there is the late phase. Okay, and in the late phase, what happens is the same orders that the mast cells released, the same molecules that the mast cells released which cause the immediate phase, which is the bronchoconstriction, they also cause a bigger inflammatory response, basically. And in the late phase, you start getting more and more white blood cells coming into the lamina propria, and basically it redoubles the entire pathological process which causes the airway hyperresponsiveness. And what I mean by that is, in the late phase, you start bringing in more T helper 2 cells, more eosinophils, okay, and therefore you redouble your attack on the lamina propria, and therefore you just make everything worse. You make the airway hyperresponsiveness worse than it already was. Okay, you make the chance that you're going to get another asthmatic attack in the future even worse. Okay, so we're initially going to discuss the immediate phase, and then we'll discuss the late phase. Okay, right. So now what we want to discuss then is the activation of these mast cells in much more detail. So let me go over the page and then we'll discuss this. So we have discussed that in both non-atopic and atopic asthma, you get asthmatic attacks in response to these non-allergic stimuli, okay, such as cold air and noxious gases. And we've discussed that that's because you activate the mast cells. Okay, and I'll start by drawing out a picture of our mast cell, because that's the central thing that we need in this discussion. Okay, and when you activate those mast cells, we've discussed that those mast cells now start screaming instructions to the bronchial smooth muscle cells, and we've discussed that one of the molecular correlates of those instructions is histamine. Okay, they're going to degranulate and release these histamine granules, and the histamine is then going to go and act on the bronchial smooth muscle cells and cause it to contract. However, the histamine is not the only molecular instruction that the mast cells release. Okay, so I'll just label this cell up as a mast cell here. Okay, the other molecular instruction that they release are molecules called cystenyl leukotrienes. Okay, and these are also capable of causing contraction of the bronchial smooth muscle cells. They're very, very capable, in fact, of causing contraction of bronchial smooth muscle cells, and they're just as important, if not more important, than the histamine, so don't neglect them. And we will be looking at these molecules in much more detail later on. Okay, right, so fundamentally then, asthmatic attacks are absolutely all about mast cells. Okay, the airway hyperresponsiveness is all about just making it more likely that mast cells are going to react to certain stimuli. Okay, and also, of course, about making the airway smooth muscle cell layer thicker so that when the mast cells do react, they have a bigger jurisdiction, basically, they have a bigger effect in producing bronchoconstriction. Okay, right, so these are the molecules that these mast cells release upon being activated, and I'll just put onto this diagram the different things that can activate them. So in non-atopic asthma and atopic asthma, things like cold air and noxious gases can activate the mast cells. In atopic asthma only, you can then get the allergen actually activating the mast cells. Okay, now, what I want to discuss then is the pathway by which you actually activate the mast cells in much more detail. Now, the pathway is not very well understood at all for cold air and noxious gases, but it is extremely well understood for how the allergen binding to the IgE mounted on an Fc epsilon R1 receptor triggers the activation of mast cells. So we are now going to discuss how the allergen can trigger activation of mast cells. Now note, this is only for atopic asthma, but most cases of asthma are atopic asthma. Okay, so this has a large amount of relevance, basically. Okay, right, so let's now discuss then how the allergen binding to the IgE mounted on the Fc epsilon R1 actually triggers the activation of the mast cell, which means the degranulation of these histamine granules, and that's another very important word here. Okay, so we saw this terminology with regards to the eosinophils earlier, but degranulation is a term that's often applied for the mast cell release of histamine. Okay, and how it results in the production of the cysteine-albuquertrienes. Okay, right, so I'm going to start off by showing you the structure of the Fc epsilon R1 receptor in much more detail, basically. Okay, so the Fc epsilon R1 receptor, this receptor that is on the surface of the mast cells, which combines to the fragment crystallizable of IgE molecules, it is not just one protein, basically. Instead, it is actually a tetramer of 4 proteins. Let me now show it. Okay, so these two parallel lines here are going to represent the inner leaflet of the phospholipid bilayer of the mast cell and the outer leaflet of the phospholipid bilayer of the mast cell. Okay, and this side is the cytoplasm, and this side is the extracellular fluid. Okay, right. So the Fc epsilon R1 receptor is made out of 4 proteins bound together. Now, 2 of the proteins are identical. Okay, so there are only actually 3 different types of subunits used to build the Fc epsilon R1. Now, the first and main one is what's known as the Fc epsilon R1 alpha, and this is the one that actually binds to the IgE molecule. So if I show this here, okay, here is our IgE molecule binding to the Fc epsilon R1 alpha. Okay, and I'll just have to squash the IgE and then bits there. Okay, so this protein here is called the Fc epsilon R1 alpha. So let's just colour everything in, okay, otherwise this picture will get very confusing. So here is the Fc epsilon R1 alpha, a protein that is a single membrane spanning protein. It's got its amino terminus extracellularly and its C terminus intracellularly here. Okay, and here now is our IgE molecule that is going to be directed against a certain epitope of our allergen. Okay, so again, I will stress we are assuming that this is atopic asthma now. In non-atopic asthma, obviously this is irrelevant because you don't have the IgE molecules against the epitopes of the allergen. Okay, however, as I say, this pathway is only really understood for how the allergen binding to the IgE, which is bound to the Fc epsilon R1, can trigger activation of mast cells. Okay, right, So there's the Fc epsilon R1 alpha. Okay, but there are 3 other proteins making up the Fc epsilon R1. So let's now show those. Okay, so the second protein that I want to show is the Fc epsilon R1 beta, which we'll have here. So this is Fc epsilon R1 beta, and I'll colour Fc epsilon R1 beta now in blue here. Okay, and both its N-terminal and its C-terminal are intracellularly. Okay, here's the C-terminal, here's the N-terminal. Okay, right. It doesn't bind the Fc epsilon R1, but it's very important in the signal transduction from the activated IgE with the allergen bound to intracellular cascades, basically. Okay, right. Then to complete the Fc epsilon R1 up, you need 2 more proteins here, which are both identical, okay? And they both have their amino terminuses extracellularly and their C terminuses intracellularly. Okay, and these are the Fc epsilon R1 gamma proteins. Okay, and we'll colour in the Fc epsilon R1 gamma proteins in turquoise here. So this tetramer of 4 proteins, 1 Fc epsilon R1 alpha, which binds to the IgE molecule up here, 1 Fc epsilon R1 beta, which is then bound to the Fc epsilon R1 alpha, and then these 2 Fc epsilon R1 gammas. This makes the full Fc epsilon R1 receptor. Okay, right. So here we have the Fc epsilon R1 receptor with the IgE molecule directed against a certain epitope of our allergen mounted on it. Okay, what we now want to see is how, when you expose this mast cell to the allergen, how is that actually going to result in the activation of the Fc epsilon R1 receptors and then the production of an intracellular signaling cascade which will activate the mast cell, okay, to release histamine and also to produce the cysteine leukotrienes. Okay, so basically what's going to happen is when you expose these Fc epsilon R1 receptors with the mounted IgE, okay, to the allergen, you end up getting cross-linking of Fc epsilon R1 receptors. So let me show you what I mean by this. Okay, and unfortunately I'm going to have to draw the picture out again because of the way I've drawn that one. Okay, so once again, here is the cell membrane, like so. Okay, now I'm going to need more than 1 Fc epsilon R1. Okay, so I'll draw these quickly. Here is 1 Fc epsilon R1 receptor, and I'll abandon showing the N and C terminals. I'll just show the transmembrane helices. Okay, and then I'm going to have another Fc epsilon R1 receptor here. Okay, like so. So I'll colour these portions in first. So here is the Fc epsilon R1 alpha proteins in green there. Okay. Here is the Fc epsilon R1 beta proteins here in blue. Okay, and then we'll have the Fc epsilon R1 gamma proteins here in turquoise. Right, now both of these Fc epsilon R1 receptors will have IgE molecules bound to them. Okay, so here is an IgE molecule bound to this one, like so, and here is an IgE molecule bound to this one. Now let's say both of these IgE molecules are directed against the allergen. However, they're directed against different epitopes of the allergen. Okay, so they bind to different portions of the allergen. The allergen, after all, could be a massive, great protein. Okay, it could have loads of different sites that the antibody molecules could bind to. Okay, so the idea here is that you've got different IgE molecules that have been produced by different B cells that have different variable regions and therefore are capable of binding to different epitopes of our allergen. Okay, but they are mounted on our Fc epsilon R1 receptors because both of them are IgE type antibody molecules. Okay, and this then means that you can get this cross-linking of Fc epsilon R1 receptors, okay, and this is really important for the actual activation of intracellular signaling cascades by these Fc epsilon R1 receptors. Okay, so once you get cross-linking like this of Fc epsilon R1 receptors, what's believed to happen is this causes the relocation of these cross-linked Fc epsilon R1 receptors. And I should stress that it's not necessary that you're only going to get 2 of these, you could get more than 2. Okay, this allergen has potentially a huge number of different epitopes, so you could have more Fc epsilon R1 receptors with IgE molecules mounted on them that could bind to different epitopes of the allergen still, and therefore you could end up with huge cross-links basically. You also have to note that this could now bind to another allergen molecule here, and then you could have another Fc epsilon R1 here linking onto that allergen molecule, so you could end up with huge cross-linked networks, and you most likely do. However, for simplicity here, we've only shown 2 of these. Okay, so end up with these cross-linked Fc epsilon R1 receptors. Okay, now what happens is the Fc epsilon R1 receptors are believed to relocate to special portions of the plasma membrane called lipid rafts. Okay, so I'll go through lipid rafts up here, I think. So basically, the plasma membrane is not homogeneous. Okay, there are certain portions of the cell membrane that are special, and these are called lipid rafts. Okay, and to crudely show what is meant by a lipid raft, let's say that we are looking at a portion of membrane here of the cell. Now most of it is just normal membrane, but then you might have these sort of areas which look like rafts, okay, which have very different types of lipids within them. Okay, and these different types of lipids tend to aggregate together into one section of the plasma membrane, okay, and that is what is known as a lipid raft. Okay, so these lipid rafts, what sort of special lipids do they actually contain? Well, one of the things that they contain a lot of is cholesterol. Okay, they also contain a lot of sphingolipids. Okay, which are lipid molecules which are based on the molecule sphingosine, okay, rather than the molecule glycerol. Okay, so most of the normal old boring old phospholipids around the outside of the lipid raft, those are phospholipids that are based on glycerol molecules. Okay, but you can also have phospholipids that are based on sphingosine. Okay, and more general than the actual phospholipids, you've got these sphingolipids, which are basically just lipid molecules that are based on sphingosine. But you can then have phosphosphingolipids, which will have phosphate groups in and therefore are also considered phospholipids. Okay, so basically these lipid rafts, they are these special areas of the plasma membrane where you have different types of lipid molecule making up these areas than other portions of the surrounding plasma membrane, which mainly consist of phospholipids that are based on glycerol and are called phosphoglycerolipids. Okay, right. Now the idea then is that once the Fc epsilon R1 receptors are cross-linked like this, they now relocate into one of these special regions of the plasma membrane, one of these lipid rafts. Okay, and in the lipid raft, they are then going to activate certain special enzymes that can then phosphorylate a protein that is present in lipid rafts. Okay, so firstly let me tell you about this protein that is specifically located in lipid rafts, which is important in the signal transduction. Okay, so I'll draw this here. Okay, and because this membrane now is lipid raft membrane, I will colour it in yellow here. Okay, so there is a very important protein that is localized to lipid rafts, and it's a transmembrane protein, like so. Okay, and this protein is known as the LAT. Okay, and the LAT here stands for the linker for the activation of T cells. Okay, so this is the linker, that's the L, and then it's for the, and then the A is for activation of, and then the T is for T cells. So it's the linker for the activation of T cells. Now, why is it called that? Well, it's because this protein is extremely important in many downstream pathways of immunological receptors. Okay, and one of the pathways that's really important in is the pathway downstream of the T cell receptor. Okay, which is why it's called the linker for the activation of T cells. Okay, right. So this linker for the activation of T cells protein is present within these special portions of the plasma membrane called lipid rafts. Okay, and that's why the Fc epsilon R1s, once they're cross-linked, need to relocate to these lipid rafts so that they can actually have an effect on these linker for the activation of T cell proteins. Okay, so in ways that are not particularly well understood at present, the Fc epsilon R1 cross-linked structure, okay, is now capable of activating certain special enzymes which are capable of phosphorylating tyrosine residues within the linker for the activation of T cells. Okay, and these enzymes are obviously tyrosine kinases. Okay, and they're tyrosine kinases that are non-receptor tyrosine kinases, so they're not attached to receptors, instead they are just free. But I think we'll call it there for this video, and we'll continue this discussion in the next video where we'll look at these non-receptor tyrosine kinases which are capable of being activated by the Fc epsilon R1 receptors cross-linked together, and which then will phosphorylate tyrosine residues on the linker for the activation of T cells.